All right, so I'm going to review the Inheritance Mendel stuff that we did in class today, just in case you need to hear it again. So Gregor Mendel wanted to be a scientist, but his parents couldn't afford college, and so he became a monk instead. Makes total sense. And he worked in the garden doing lots of science at the monastery. He studied pea plants, so Mendel was all about the peas. Before we start talking about inheritance, let's go ahead and review some of that vocab. Remember that gametes are sex cells, eggs and sperm. They're haploid. They only have half the number of chromosomes as other cells do. Zygotes are going to be the resulting cell from the fusion of egg and sperm. So these will be the first cells that go back to being diploid or having that regular number of cells. Not regular number of chromosomes. Cells with a regular number of chromosomes. Segregation of alleles. This is just that idea that when we're building eggs and sperm, we don't shuffle all the stuff that we got from grandma onto one egg and all the stuff we got from grandpa onto the other egg, but in fact it gets mixed up magically. Fertilization is also going to allow for recombinations of alleles, which leads to genetic variation, which is really good for population. Remember that dominant, recessive, dominant traits, dominant alleles are going to mask the expression of a recessive trait. Homozygous, you have two of the same, whether dominant or recessive. Heterozygous, you have one of each. And then genotype and phenotype. Genotype, these are the alleles that you have. We could show that with letters. The genotype could be AA or AA or AA. And then your phenotype would be your earlobes are free or your earlobes are attached to the side of your face. So phenotype, that physical trait that you can see. Genotype, the alleles that you get for that trait. So Mendel's, the traits in pea plants that Mendel looked at, that he researched, were interestingly all simple complete dominance, which is pretty uncommon. He probably chose the traits on purpose because they were doing what he expected, as opposed to Morgan later with his linked genes in the Drosophila. So we had that complete dominance, and we can show Punnett squares to predict what the offspring will look like. So if tall is dominant, I've got a homozygous tall plant. That's going to be big T, big T. Short plant has got to be homozygous recessive. And then I can do big T, little t, little t, draw my Punnett square, and 100% of the offspring are heterozygous. Sometimes we draw these guys on an angle. Same deal though, big T, little t, little t. And when I mush them together, we get all heterozygous. Easy peasy. So let's say that I have a tall plant, but I don't know if it's homozygous dominant, big T, or heterozygous, big T, little t. I don't know. Both of those are going to have the phenotype of being tall. How do I figure out if it is homozygous dominant or heterozygous? I'm going to do what's called a test cross. And I mean, a test cross always happens with a homozygous recessive individual. And here's the magic. If I have a homozygous dominant parent and I cross it with a homozygous recessive parent, then 100% of the offspring will show the dominant trait. But if my parent was in fact heterozygous and I cross that parent with my homozygous recessive test parent, it's odd, test parent, then I should have 50% with the dominant trait and 50% with that recessive phenotype. And so with that test cross using homozygous recessive, I can figure out for sure, well, I mean, statistically, I can figure out if I've got a dominant, homozygous dominant parent or a heterozygous parent. Good. So we talked about mono versus di. Mono means one, di means two. So in this instance, I'm looking at a plant that is tall and has green seeds, heterozygous tall and homozygous dominant green seeds. It's crossed with a short plant, must be little t heterozygous seeds, big G, little g. So to do this cross, I need to build egg and sperm. Let's say that this is dad. I need to build some sperm from him. And each sperm is going to have one allele of each gene. So I could have a sperm that's big T and big G, or I could have a sperm that's little t and big G. And that's about all the combinations that I've got there. If I'm looking at this mama plant and eggs, I could have little t with a big G egg, or I could have little t with a little g egg. And that's it. Those are the only possible combinations of T's and G's. So that I can build my Punnett square. There's dad. Here's mom. And the offspring are going to look like this. So I have heterozygous tall, homozygous dominant green. So tall and green. And then I can have short and green, homozygous and homozygous. And then I have here another heterozygous tall and a heterozygous green. So still tall and green. So now I've got two of those guys. And here I've got short and green. So another short and green. So 50% tall and green, 50% short and green should be the resulting offspring. Codominance is a quirky idea in which alleles are not dominant, or they're all dominant. It's hard to tell. And we have a couple different ways that this can be shown. So in this instance, I've got a red flower and a white flower, and neither one is going to win out over the other. The individuals are all a blend of red and white. And so all of my heterozygous individuals here are pink flowers. So my chickens down here, I've got a white chicken and I've got a black chicken. And rather than having white or black or gray, we have a chicken that is both white and black. So he gets a little bit of both of those colors of feathers instead of a blend of the two colors. So these are both examples of codominance. Sometimes we call it incomplete dominance. And so both phenotypes are shown in the offspring. We humans have some traits that are codominant. One example is human blood type, which is interestingly both codominant and normal dominant. So there are 3 alleles instead of only 2. So instead of being big A and little a, we have big I and little I, but we have 2 different versions of that dominant I. We have A's and B's. So I can look at putting together a cross. A person who has type O blood is going to be homozygous recessive, so little I, little I. We're going to cross that with a person who has type AB blood, which means that they have both A and B, so IA, IB. We build our Punnett square, I, A, B, and we get IA, little I, little I, B, little I, B, little I. So 50% of the children from this family are going to have type A, and the other, I forgot the A, the other 50% are going to be type B, which is weird because neither mom nor dad had type A or type B. So we can see how those alleles are recombining. So now we need to figure out with blood type if this suspected guy could be the daddy. So mom is type A, so she's either IA or IA little I. And baby's type A, so again could be this or this. And the potential dad has type O. So dad has to be little I. So could this guy be the daddy? And the answer is yes, he could be. We don't know for sure, but we know that he could be. Because mom, whether she is homozygous or heterozygous, is going to with that individual with type O blood, be able to make babies that are also type A blood. So what about this instance? What if mama is O and